Now, it is my great pleasure to introduce Dr. Karen Liddle. Karen is a senior staff specialist paediatrician at the Queensland Children's Hospital and PhD candidate with the University of Queensland with the Integrating Genomics into Medicine group. She has been clinically and developmentally assessing children with permanent hearing loss in the Childhood Hearing Clinic for 13 years. Her current research engages national and international collaborators to evaluate the utility of genetic testing and imaging in informing diagnoses and improving outcomes in childhood hearing loss. So I'll now hand you over to Karen.
Thank you very much for having me today. I'd also like to pay respects to the traditional custodians of the lands in which we work, talk and live. I'm coming from Meanjin, and it's the Yagara and Turrbal people here, and I'd like to pay respects to all the country where everyone else is dialing in from today. And I'd also like to pay respect to all the deaf community and from the different areas that we're dialing in from today as well. So I work at the Queensland Children's Hospital in Brisbane, and I'm just doing this webinar today. Anne asked me if I would share it. It's based on a presentation I gave at the Australasian Newborn Hearing Screening Conference in Hobart earlier this year. And it is looking at some work that we've done and also an update about different changes that are coming with different access to genetic testing. So it's whatever attitude or if you do want this, if you don't want this, it's good to be able to have the background information to be able to make an informed choice. So I'm going to go back to some of the basics. A lot of this, people might not have seen this since high school. So, and this field is really advancing at quite a fast rate. So I'm just going to explain some of the background about what some of the tests are based in, and so to try and help make a bit more clear what we're talking about. So when we're talking about genetics, we're looking at, so that's the instruction for our body. So in the cell, in the nucleus of our cells, there's the chromosomes, and that's all this tightly wound DNA, which you can see there in that picture. And the DNA is made up of nucleotides, and they provide instructions for proteins, and that's what makes up our bodies. And when we're thinking about genetic conditions, there's three main categories of genetic conditions. So there's conditions related to the chromosomes that we were just looking at. There's conditions called monogenic, and that's related to single genes. And then there's what's called multifactorial. So genetics plays a role, but there's other environmental impacts as well. And sometimes it's lots of different genes together. So just to look at that a little bit more, when we're looking at chromosomal changes, so that can be, they call copy number variants. So it's the change in the number. So sometimes there's we normally have 2 copies of the chromosomes in each gene. So you can have 3 copies, such as trisomy 21, which is Down syndrome. And that can be a whole chromosome, or it can be small parts of chromosomes. So extra bits or missing bits that can be quite small, but it's parts of chromosomes. So that's usually a number of genes together. Then you can look at what they call the monogenic. So it's single gene, and that's just sometimes a change in a single base pair in the DNA. So quite small. And that's conditions such as cystic fibrosis and muscular dystrophy. And also there's different patterns of inheritance related to deafness as well. And then the multifactorial is genetic and environmental factors. And commonly known things are heart disease, diabetes, cancer, kidney disease, where there's lots of different factors impacting, but genetics are playing a role. But genetics also has information such as for our blood pressure, height, hair color, all that kind of thing is that multifactorial type of inheritance. And so this is just another figure looking at that. So we've got an interaction between genetic factors and environmental factors for those multifactorial types of inheritance. So how does this fit with deafness? So when we're looking at causes of deafness and hearing loss, when we're talking about, if you look at that green half of the chart there, that's what we're talking about, your chromosomal and monogenic type inheritance. And then the other half is that environmental. So we've got some conditions that are environmental, such as CMV or being exposed to chemotherapy agents, things like that. But then there's also factors where there's genetic factors playing a role, as well as environmental exposure, such as noise and things like that. And then when we're looking at this green side, we can see that is broken up into what's called syndromic, where there can be other things inherited with deafness, and then non-syndromic, where deafness is the only thing caused by that genetic change. So when we're looking at our basic genetics, so this is a picture of what's called the carrier type. So that's all the chromosomes that they can be sort of seen under the microscope like that. And so you can see there's 23 pairs there, and you get half from your mum, half from your dad. And the changes in those used to be called mutations, but these days people call them variants. And the genotype is what the changes are and how that's written about the changes in the DNA. But the phenotype is what that looks like. So that's in an individual. It can be physical changes, but it also can be related. So not everybody with the same genetic change has the same impact on them. So that's what we call phenotype. So that's how it presents in an individual. So those are some of the words that we'll be using. And so, yeah, the commonest non-syndromic genetic cause of hearing loss is what's called connexin 26, or the gene for that is called GJB2, and that's there on chromosome 13. So people may well have heard about this one. And so this is related to non-syndromic hearing loss, and it can cause 20 to 50% of the non-syndromic group. So it is quite common. And that's usually, if people are having genetic testing for hearing loss, that's usually where we start, unless there's other physical clues pointing towards something else. And so the way that is inherited is in a recessive way. So I don't know if you remember from high school, but there's different ways that things can be inherited, and recessive usually means you need two abnormal copies of the gene for it to show up. So one from each parent usually is how that works. And a parent then with one copy of the gene is called a carrier. And so for people, when they're having children, if they're both carriers, there's a 1 in 4 chance that they'll have a child who inherits both copies of the variant gene. And there's a 1 in 4 chance that a child will inherit both normal copies, and then there's 2 in 4 chance that children will be carriers. And so that is every time you have a pregnancy, that is the same, regardless of what children you've had before. And the way then we talk about as well, homozygous means the same copy of a gene on each chromosome, and compound heterozygous means a different variant. They're both variants that can cause deafness, but they're different from each parent. So, and that's called compound heterozygous, and heterozygous just meaning different on each chromosome. And so on that picture there, that shows you if you just have one change in the gene, that's called heterozygous, and that's being a carrier. And then homozygous, the same on each chromosome. Compound heterozygous is one on each, but different. So when we're looking at hearing loss related to connexin 26, 98% or more are inherited in that autosomal recessive way. But there's a small amount that can be inherited in a dominant way, which means only one change is needed. And that's usually in each generation in that case. And there's another gene, just to make it complicated, right next to there called GJB6 or connexin 30 that can also be inherited with connexin 26 and have the same effect. So if we're looking at, that's our connexin 26, if you've got a copy on each chromosome. And then this is with the connexin 30. What our lab normally does is if they look for this gene and they find one variant copy in a child, they will then look at connexin 30 to see if there's any changes there. So that's a lot of background about that type of genetic testing, which is the basic testing that we do. And then this is a picture that's probably familiar to people, I think. So looking at, the green is a section through the cochlea, and then we've got enlarged there at the bottom, the organ of Corti, which is the sensory organ for hearing. And that's just to orient you, because the next slide I've got is a picture of where the connexin 26 gene is expressed. And so if we have a look, that's that same picture we were just looking at, but you can see the green fluorescence. That's where that GJB2 or connexin 26 gene is expressed. So it's right through the organ of Corti, and it's involved in potassium recycling there. So sort of at that cellular level, and it's in the supporting cells. And then this is a picture of what it would look like with two cells next to each other there in the organ of Corti. And so you can see the purple is what connexin 26, you get 6 of those, and they make up this channel, and that's how the cells communicate next to each other, and potassium flows across that there. So that's exactly what that gene is involved in doing and where it is. So that's some more background then, I guess, about where that is, and that's usually then just hearing loss. So this is some data now that, because that gene was discovered in 1997, it's been tested since that time, and having a look about around the world, which genetic changes are the most common in the different countries. And this was published 10 years ago, this data. They pulled together all the different information from around the world, and you can see the blue is one change called 35del-G. So that's a variant that's most common in people from European backgrounds. And then you can see the green, which is in China, and that's a different variant there, 235del-C. That's the commonest one there. And then the yellow, that's V37I, that is Southeast Asia. So that's a common gene that's seen there. And these ones, so there's carriers, when they look at the countries, how common is it for people to be carriers? It's between 1% and 2% for the top two changes, the blue and the green. But that yellow in Southeast Asia, it's 18% of the population are carriers, which is actually very common. And that one's associated with a milder hearing loss. So that's just looking at Australia there. That's what's been published at that time. And this is from another paper, but that was very similar, looking at the data. And this one is showing that this is from the Lockie study, which people might be familiar with through Hearing Australia, have followed up some children. This was published in 2013, and there were 82 children that they looked at there. This is what's been pulled into this paper. And the commonest change was that blue was the same blue from the previous slide, which was that 35del-G. And you can see the green, which is the next most common. That's that Southeast Asian one that was yellow in the previous slide, that milder variant. So that is what is in the literature. This is 10 years old now, but that's what was the most common thing. So that's what we expect to see in Australia. And then there's a little bit more been published from the group from Victoria, and they had a smaller number, but they were just looking at 35del-G and other in that group. So that's the data from Australia that's been published. So for us in our clinic, we looked at all that, and we thought, well, there's variable ways that people can present with the different genetic changes, but there's not been a lot published from Australia. And we thought that if we had more data about that, we would be able to help talk to families about what they're going to expect into the future, and also make decisions about management as well. So what we did then was audit our clinic. So we wanted to describe children from Queensland who'd had this investigation for bilateral sensorineural hearing loss, so that's both sides, to assess the frequency of the GJB2 or common GJB6 variants, and then look at our clinical data to see that genotype-phenotype correlation. So what does that show? Do we see severe hearing losses or not? So that was what we were hoping to have, some more local data instead of having to rely on small numbers or data from overseas. So this was a study then, retrospective, so we're looking back and just looking at the charts basically. And so these are kids who were seen through Queensland Health, ENT and or pediatrics clinics, and who had that connexin 26 testing performed through Pathology Queensland. And this was between 2014 and 2019. And then we went to the charts and looked at audiology reports, other medical records, information from our statewide database, our hearing screening database, and then put all that into a spreadsheet. And the things we were looking at in the chart, we wanted to know, when we're talking about this audiological phenotype, so it's like, what type of hearing loss do you have with the different genetic variants in that gene? So we were looking at degree of hearing loss, and that was in the better hearing ear, was how we were describing that one. We were looking at symmetry, so were the sides each ear the same or different? Was it stable over time or fluctuating or progressive? And what was the shape of the audiogram? Other things that would be nice to look at, but that we didn't collect in this study, would be, I think, speech and language development, and potentially cognitive and other development. And I'd be certainly interested to get feedback from you, are there other things that you think are important that we should be thinking about and monitoring over time? So in terms of our study, there were 625 people in that 5-year, children in that 5-year period who had the testing. And there were 490 that didn't show any changes in that gene. But there were 135 who did. And then when we went to look at the charts of those children, there were 8 who had other causes of their hearing loss. So 3 had absent cochlear nerves, 2 had large vestibular aqueduct, and then there were 3 with other genetic changes that we thought better explained their hearing loss, like Waardenburg syndrome, and then a couple of other deletions on the chromosome there. So then we had that left us with 127 charts to audit. And then over time, when we were looking to see the initial audiograms and then follow up, we had 103 people who had information over more than one time point. And so this is out of those 127 children, 72 were males and 55 females. And the average age at testing was 1.8 years. But when you look at this graph here of age, the median age was actually 4 months. So we can see that there's a big spike there at 4 months of age, and 2/3 of people had had the testing under 12 months of age. So that's really reflecting people who had hearing loss picked up on the newborn hearing screen. And some of the other kids either elected to have testing later or moved here from another place, or their hearing loss was diagnosed at an older age. And then when we looked at some of the, so the degree of hearing loss in the better ear and the latest. So the better ear is the initial is the green, and the latest is the orange. And so when we're looking across the x-axis there, we can see, so we've got normal, which because it's the better hearing ear, some people had unilateral hearing loss, and so that's then coded as normal. but you can see that there's a few there. Mild was the commonest, and this was a bit of a surprise to us. And then you can see the others: moderate, moderately severe, severe, profound, and there's a little bit of missing data. And I've just put a red box there, and that's what around moderate or worse is what the hearing screen is designed to pick up. So our hearing screen actually picks up milder hearing losses than what it is aiming to. And so we thought, well, why are we getting people with this normal result? We had 25 children that had a normal result in the initial ear. So I had a look closer at that group. There were 12 that had unilateral, so they did have a hearing loss, but it was one side. We had 7 in the pink there, that's minimal or slight. So that means you didn't, for the mild, you had to have a 4 frequency average. So these might have had like a one frequency with what's a mild threshold. So that doesn't code as mild in this study. So I call that minimal or slight at that time. We had 2 with temporary conductive, and that can be hard to tease out sometimes in a baby. And then 4 that had actually normal hearing at that time. And then the better people who had normal hearing ear in the latest audiogram, there was 16, and we saw still some with unilateral, but you can see that some of those unilaterals had progressed to bilateral. And we still had the minimal and slight, the temporary conductive, and 4 with normal, but that was actually not all necessarily the same people as in that original one. So some people had fluctuating hearing. This is the slide that's a lot there. What I want you to take away from this one is this is just the different variants that we saw and then the number of them. And so you can see there that does this come in? The commonest was this V37I, which is that Southeast Asian variant. That one was in the yellow back on that world map. That was our commonest, which is not what we expected. But then the second most common was that 35 del G, which is the European one. And then there was a number of others, but a lot of the others were in small numbers. So that's just a pie graph showing that the relative proportions of those genes there. But then we start to say, okay, then how were they inherited? Remember, we talked about homozygous, heterozygous, those different ways. We can see that we had one child with dominant hearing loss, and then we had, this was our homozygous group, so 2 of the same variants each. Compound heterozygous was different in each chromosome. Heterozygous means you had one variant gene, one normal gene. And then these last ones there was a change, but we're not sure if it's one that actually causes any hearing loss. Looking at whether they progressed or not, we had majority were stable. It was only over sort of a 5 to 7 year period that we had that data. Some were progressive and a couple were fluctuating. So that's, and looking at it by inheritance pattern there. So majority were stable and the homozygous were mostly stable. And then in terms of those other audiogram characteristics that we had, there wasn't really a lot of evidence of association between your genetic change and either the symmetry and stability of impairment there. And because we had small numbers in some of the groups. But when we're looking at the homozygous group, there were 45 in that group, and we had only 3 variants. So the most common 3 variants were homozygous. So we had our 35 del G, the European one. We had the Southeast Asian one, and that had significantly more mild at initial and latest. And this is showing you what those graphs look like. If you look at on the left, you can see the homozygous of that. This is our Southeast Asian variant there, and it's either normal, mild, moderate. Nobody was worse than moderate in that group. And then the ones that was the 35 del G had the more severe changes. So they had some profound, severe, moderate, and moderately severe there. So it was a little bit unexpected to find that one's the most common, but that's really useful information for us when we're talking to families about it. And particularly that it was associated with mostly mild hearing profiles. And it's also showing that some of the milder hearing loss that's getting picked up does have a genetic cause. So I was just going to talk through a couple of cases really briefly to sort of illustrate what this looks like in the clinic, because I know that that's a lot of data that I've just shown you that's probably a little bit dry. So this is baby Miles, and he had a bilateral refer on his newborn hearing screen. So both sides tested as refer. And then when we went for audiology testing, he had bilateral moderately severe sensory neural hearing loss confirmed, and had a normal physical examination. And there was no family history of hearing loss at all. And he had an MRI of his brain, and the hearing structures were normal, as was the rest of the brain. But then we did the GJB2, or our connexin 26 testing, and he had compound heterozygous variants. I think one was a 35 del G, and one was, I think, our third most common variant. And so that explains the hearing loss for them. And I guess there's information for his parents when they're having more children. There's a 1 in 4 chance of having a baby with a hearing loss next time, because they were both confirmed to be carriers. So then they had testing to look and see, did they carry any of the variants? And yes, they each did carry one. And then the other patient I was going to talk to you about was baby Lucy. And this is something that's happening a bit more commonly now, but her parents had prenatal carrier screening. So they had testing before a pregnancy to see, did they have any, did they carry any genetic changes that to be aware of? And they were both found to be carriers of the V37I variant. So that Southeast Asian mild variant. And they both were from Southeast Asia originally. So they knew that already. And this is something that people would pay for privately, sort of through either an obstetrician, or sometimes people will send samples overseas. And Lucy had a unilateral refer result on the newborn screen, and her audiology showed a slight / mild hearing loss at 4 kilohertz. So that's a high frequency in the left ear only. So she would have been coded as normal in our study. And usually if I'm seeing a baby with a hearing loss like this, I would usually wait till the next hearing loss at 9 months and see what happens. Because to see, sometimes the testing can be a little bit tricky early on. And if that's that one frequency and mild, we're not really sure sort of what that meaning of that is long term. But her parents were keen to know, could it be related to their genetic results? And they really wanted to have her tested to see, did she have those variants or not? And she did. So she was homozygous, so had a copy on each chromosome of that V37I. So that's inherited one copy from each parent and had this very, very minimal hearing loss at this age. And one of the things that's helpful when we've got that specific information about that gene is that not everybody who has that change will show up a problem. So they call that incomplete penetrance. So not everybody who has the gene change shows up a problem. So and it also can be fluctuating over time. So she needs to monitor, and we support her communication, but to see what happens over time. And I think for them having more children, it'd be reassuring. So in terms of future directions, in our study, we found that where I've put in the red there's quite a few people that had hearing loss that only had one change in their connexin 26 gene. And so it's potentially related to other genes or other factors, their hearing loss. But we haven't really had access to more detailed genetic testing easily in Australia. I need to refer to our clinical genetics service in Queensland. It takes a year to get in there, and sometimes they'll fund testing, sometimes not. So that is that's how it has been lately. I'm just going to talk to you next, though, about some changes that are coming. So I was thinking for the future considerations from our study, thinking about looking at outcomes and monitoring over time to see so that we can give better information to people about what to expect. And if people are having prenatal genetic carrier screening, we can say to them, yes, this V37I gene change is common and it was mild. We didn't see anybody with a hearing loss worse than moderate in our study. And it's, has been consistently associated with mild hearing loss. So in terms of other genetic testing, what I'm aware this is a lot of information that we're going through. So the types of genetic testing that there are, so if you've got there on the left is a single gene. So that's what we're talking about with the connexin 26. We are just asking to test that gene. They sequence that whole gene and only that. So that gives us really good information about that gene, but it doesn't tell us about anything else. The other kind of test people might have had or heard about, chromosomal microarrays, that is looking at all the DNA. And that's that chromosomal type inheritance we talked about at the start, where you can get 3 copies or 1 copy of certain parts of the chromosome. And then in terms of more detailed testing, as the technology has advanced and the cost has come down, there has then been access to testing, like there's a multi-gene panel, so that where they can all at once test, it's often around 200 genes for hearing loss, and that can be that's much quicker than just testing one at a time. And then the next level of detailed testing is the whole exome and whole genome sequencing. So I think I've got that on the next slide. Yeah, so that the whole genome is all of the genetic material. So that is a lot of data, as you can imagine. But as technology is advanced, that it is possible to sequence that, and they can do that and get results depending on lab capacity, really. And that's people to interpret the results. Can be a turnaround time of 3 to 4 months, sometimes longer. But that's actually a lot quicker than it used to be. The difference between whole genome sequencing, whole exome sequencing, exome is the coding region of our body. So the regions of the DNA that we know code for genes, that code for proteins. So that's 1 to 2%. So that's sort of, if you got you want to start looking, that's usually the best place to start looking. And this is just a way to sort of visualize what the difference between those tests. So you've got the whole genome sequencing is everything. Exome is the little portion that codes for genes. And then gene panel is just looking for genes related to a specific condition. So the difference between those type of tests is the types, understanding the type of results that you can get. So these are the type of results that you might get when you have a genetic test. So it can be positive, and so there can be a variant found that explains the condition. We can get a result that's uncertain, so that there's a variant found, but it either hasn't really been described before in terms of hearing loss, or it's not clear whether it explains the condition or not. There's not a lot of information about it, or it doesn't seem, what they know about the functional changes it might cause, it doesn't seem like it would have a big impact. So you can get a result like that. You can get a result that's negative, so that they have a look and they don't find any variants that explain it. And that's, as technology advances, that changes over time. And then you can also get results that are called incidental findings. And so that can be if there's a variant found in a gene that's not related to the condition that we're looking for. And that can be different, obviously the different types of tests that I showed you. If you're specifically looking for a gene, you're not going to be finding other things because you're only looking for that. Whereas if you cast the net wider, you are risking, I guess, finding variants in other genes that you weren't necessarily looking for. Now, in Australia, they're quite strict. And so that all of that information is there, but they're not analysing all of it. Whereas I guess if some testing is done overseas, sometimes the rules around what is reported and what is tested by laboratories can be different. And so people might end up finding information about cancer genes or other types of genes that they weren't really looking for. So that's understanding the types of testing that you can have and the types of results that you might get. So in terms of our Australian context, in terms of that more detailed genetic testing, there's one of the clinical geneticists from Melbourne, Dr. Lillian Downey, did a study, and I'm just going to briefly report the results of her study. And she's, on the back of that, been able to advocate for increased access to testing here. So her study that she did was a 2-year prospective, so looking forward cohort study. And so what they did was for children who had bilateral sensory neural hearing loss, and it was at 3 frequency, 40 decibel or worse average. So those were the people who were eligible to be included in the study. And it was over a 2-year period, 2016, 2017. And in that time, the population of Victoria, they estimated there'd be 150 babies that would be eligible for inclusion in that study if they wanted to. And what they found, so there were 106 people who opted to be in the study. And the results they had were, in terms of causes of hearing loss, the connexin 26 was 20%, which you may remember from some of the early slides. That's what we expect, between 20 and 50% in some populations, connexin 26, and that they found the 21%. And then there was 5% who had CMV as a cause of their hearing loss. And then they were able to find genetic causes for other non-syndromic deafness in 15%, and syndromic deafness in another 20%. So they were able to, these two coloured, the blue and this brighter green, were an increase in finding out a cause for people than what they had been able to before. And some of the syndromic conditions are things like Usher syndrome, where you can have progressive changes in vision over time. So there's a number of different conditions that they found in that study. And so on the back of that, they led an application to the Medicare Services Advisory Committee to be able to access Medicare funding for that type of testing. And it's been approved. So we heard that early this year, it was in March, that we heard that that's been approved. And I think from last I heard was November is when we should be able to actually access that. But it actually depends on laboratories around the country and what their costs are and if they're happy with the Medicare rebate for the testing. So it may be a little bit different depending where you are, but there will be access to Medicare funding for this type of testing. I've got a little bit more detail. This is what has been recommended in this Medicare, is that a specialist or consultant physician, so that's a geneticist, pediatrician, or it could be an ENT experienced with childhood hearing loss, can request it. That the hearing loss needs to present prior to 18 and be permanent, moderate, severe or profound, and unilateral, or bilateral, sensorineural, auditory neuropathy, or mixed. And so that's the eligibility for that testing. And then, sorry, I just read that bit out for you. And we need to have appropriate informed consent prior to the testing. So I've touched on it really briefly, but this kind of testing is very detailed. So you really do need to have a conversation. It takes about half an hour at a minimum, I think, to talk through all the pros and cons and decide whether it's right in the family context and what it can and can't tell you. So appropriate genetic counselling needs to be done pre-testing and on receipt of the results as well. And so that's got us thinking about models of care, because at the moment, genetic counsellors, what we'd really like to do is try and embed them in the clinics to be able to have better access to this testing sort of around the time of diagnosis if people do want it. Because yes, it does take, there's a lot of things that people are talking about and trying to make decisions about, and that to be said, there's no urgency on the timing of the testing. I guess we're guided by families and what they want, but we do need to have appropriate time to understand it and make those decisions. So that is all that I've got in my presentation. I'm very happy. I know that I've covered a lot, and it's I'm really happy for questions and feedback. If anything you'd like more details about, but also to say this is mainly, I'm not saying that I think everybody should have this. I think that it's an individual choice, and I think to make an informed choice, it's really good to understand about the testing and what it is, and what it can and can't tell you, so that you can then make what you feel is the right choice for your family. Oh yes, so I can see there's a question: is enlarged vestibular aqueduct a genetic hearing loss?
That was the one question we've got there.
Yeah. So that's a really good question. And yeah, what that is an appearance on the MRI, and there are a number of different genetic causes for that. So it can be related to specific genes, or it can also be related to syndrome. So other features as well can be part of different conditions. So yes, I would put that in a genetic cause, but if we find that, then we're not thinking connexin 26 is the cause of the hearing loss in that child. We're thinking that the enlarged vestibular aqueduct is. I can see a question here. It says, what kind of decisions might be made after genetic testing? Is it whether to have more children, identification of other diseases, and future management? So that's a really insightful question as well, as to say, for some, at the moment, where our management is the same in terms of we monitor hearing, we support if amplification is appropriate, people get linked in for that, and supporting communication development. So genetic testing, I guess, is can be helpful to, do we expect it to get worse or not? Do we expect other body systems to be affected? And I guess for parents, for some people, they say, it is what it is. This is not going to make any difference to decision making for us. And then other people find that they do want to know, and that, I guess they're thinking about future pregnancies and things like that. Not in Australia, there is access to reproductive carrier screening and things like that. That's done through genetic services, but a lot of people I speak to, I guess it's just being aware of a possibility, so they feel prepared. But I guess the more that we know, the more that we will be able to personalize care for people. I can see there's a couple of other questions. But in terms of future management too, I guess as technology advances, I think, if that will probably change, offering more personalized care for people. So my son has mild to moderate bilateral sensorineural hearing loss, connexin and CMV were negative. Pediatrician recommended not doing any MRI or further testing until 5 years of age. However, ENT have said they would like to complete the MRI to look for a cause. I'm unsure what to do next. Who would be the best person to go to now? So I guess when we're looking at that, I guess MRI, when we're looking at it, it's potentially, I'm wondering if they were thinking about you'd need a sedation likely until before age of 5, and it gets safer the older kids get. So we rather not do them in babies if we don't need to. But if school age, if kids can lie still, they can have an MRI. There's no radiation from it and not needing an anesthetic for it. But however, if the hearing is deteriorating, it is really helpful to get one. And I think that's probably based on whether it's going to change. Is it going to change management or not? If your hearing is stable, it's probably not going to change a lot. And I'm wondering if that's what they're weighing up in that situation. So we would be monitoring hearing over time, but certainly there will be access to this more detailed genetic testing as well, if that's something that you're interested in coming later in the year. But yeah, so those are the things that could be done. And I guess the MRI can show things like we discussed, enlarged vestibular aqueduct is a change that we can see on the MRI. And that can be helpful to know about because you need to avoid contact sports if you have that, because you can have progressive hearing loss with head trauma. So that's why it's good to have an MRI at some point. But obviously in little kids, they're not usually, playing contact sport, doing scuba diving, that type of thing. And I can see another question there. My daughter has a connexin 26 gene and it wasn't picked up on the newborn screen. We only found out about 4.5 years of age. She is currently mild to moderate bilateral. Our biggest concern is it going to get worse or stay at this level? Yeah. Oh, so this study, I guess our study isn't powered. So the question is it, am I right to say this study indicated it should stay the same? So we didn't follow people right up all the way through into adolescence. And what we found, I guess, is we had more detail about that milder variant. So the information can depend on the specific variant that your daughter has. But yeah, I guess it is hard. It is hard to say. And I probably didn't mention, but it is so complex that even in siblings, they can have a different hearing loss because there can be other factors that are interacting that we're not sure of because the genetics are really complicated. So yeah, it's one that it can change over time. And I guess hers potentially has changed if it wasn't picked up on the newborn screen.
Okay. Thanks, Karen. That's wonderful.